Now let's take a closer look at exotoxins and how they work. Most often, the exotoxins are produced by gram-positive bacteria. Here the example is given. We're going to use Clostridium botulinum producing Botox, botulinum toxin. And the bacteria, they will produce the toxin inside. Most often it's called an AB toxin. We'll take a closer look at that in a minute. And then the toxin, which is a protein, is secreted to the outside of the cell, where it then binds to host cells and causes damage. So most of the time we have AB toxins. That means the exotoxin has two components, an A component and a B component, where the A component is the enzyme part of the toxin, and the B part, B for binding, that's the binding component of the AB toxin. And here they're using diphtheria toxin as an example. So here let's take a look at the steps of how an AB toxin works. The bacterium produces the AB toxin. So right here we have the active and the binding portion of this toxin. Both components are proteins. And then this protein is released from the bacterium, and the B portion, the binding portion of the AB toxin, will bind to a receptor here on the host cell. So there's the receptor binding. The host cell will take the toxin inside, it will internalize it through a vesicle, and then the AB toxin is basically picked apart in the host cell and activated, and then the enzyme portion of the AB toxin causes the cellular damage. Here you'll see that the A portion, the active portion of the exotoxin, inhibits protein synthesis in this particular cell, and the binding portion of the AB toxin is actually expelled from the cell in this case. Membrane disrupting toxins, those are also exotoxins. They just don't follow this AB pattern. They're a little bit different. They lyse host cells by disrupting plasma membranes. Here are some examples given. The leukocytins, they kill phagocytic leukocytes. Hemolysins, they kill erythrocytes by forming protein channels. And streptolysins, these are hemolysins produced by streptococci. Some pathogens, they also produce what's called superantigens. They cause a very intense immune reaction that's referred to as a cytokine storm. So they release cytokines and then cause a massive reaction of the immune system that can cause fever, nausea, vomiting, diarrhea, shock, and even death. You may have heard about this new symptom that's seen in children, where the immune system overreacts to a coronavirus infection, and these kids, they're experiencing exactly this, a cytokine storm that causes all of the problems then, an intense inflammatory reaction basically. Genotoxins damage DNA, they cause mutations, they can disrupt cell division and also lead to cancer. So here is a selection of different diseases that are caused by exotoxins, and so you should definitely take a look at some of these here. The first three are very important for you to know: botulism, tetanus, diphtheria. They're all caused by AB exotoxins released from the bacteria listed here: Clostridium botulinum, Clostridium tetani, Corynebacterium diphtheriae. All of these we had in pathogen groups so far. The scalded skin syndrome, cholera, traveler's diarrhea, and anthrax, they're also all caused by AB exotoxins. Here are some membrane disrupting exotoxins that we would see in C. diff. That would be gas, gangrene, food poisoning, and then antibiotic associated diarrhea. Superantigens could be used by bacteria that produce toxic shock syndrome. That would be Staphylococcus aureus, or can also cause food poisoning. And a genotoxin example here, Helicobacter pylori, causing stomach cancer. Moving on to endotoxins. So Endotoxins, they're basically part of the cell wall of gram-negative bacteria. So specifically the lipid A portion of lipopolysaccharides of gram-negative bacteria. They're released during bacterial multiplication and more often when gram-negative bacteria die, as in they're being digested by a macrophage and then the toxin is actually released through this mechanism. They typically stimulate macrophages to release cytokines, and then they can cause disseminated intravascular coagulation. So let's take a look at endotoxins. So they're part of gram-negative cell walls, gram-negative bacteria, this lipid A portion. And here we have an example given, Salmonella. The endotoxins you can see here, they are composed of lipids that are part of the cell wall. And these endotoxins, they are liberated when the bacteria die and the cell wall lyses or breaks apart. Here we have the example of an endotoxin that causes fever, which is often the case. So here the endotoxin is part of the gram-negative cell wall. This macrophage has found this gram-negative bacterium, is engulfing it and ingesting it and digesting it. And so now it's liberating the toxin, actually, it's right here. It's liberating the toxin from the gram-negative cell wall. That toxin ends up going into the bloodstream, and then it causes the hypothalamus to reset the temperature, and ultimately that will cause fever. Other endotoxins, they might produce shock, such as septic shock, as an example. Moving on here to an assay that can detect, is very sensitive to detect the presence of endotoxins. And it's called the Limulus amebocyte lysate, or LAL assay. And basically the way it works is you're using Limulus, which is the horseshoe crab, Atlantic horseshoe crab, contains amebocytes. The amebocytes, they lyse in the presence of an endotoxin and produce a clot, and the degree to which this clot is produced is sort of this measure for how much endotoxin is present. Here's a table that summarizes exotoxins and endotoxins. Be sure to study this table. This is actually pretty important, and it's a really nicely done summary of endo versus exotoxins. So exotoxins, the bacterial source would be gram-positive bacteria, and for endotoxins we have gram-negative bacteria. An exotoxin is usually a metabolic product of a growing cell, and it's a protein, usually with these AB parts, versus the endotoxins. They're present in the lipopolysaccharide of the outer membrane, so they're part of the gram-negative cell wall, and it's usually a lipid portion from the bacterial cell wall. The pharmacology, or the effect in the body, there's various kinds, and you can review that from other slides. Heat stability, since exotoxins are proteins, they are not heat stable. The endotoxins, they can withstand higher temperatures, some of them even autoclaving at 121 degrees Celsius. The toxicity is high for the exotoxins and fairly low for the endotoxins. Fever producing you would see only in the endotoxins. And the immunology, exotoxins, they will produce antibodies. So you can use the exotoxin portions, an inactivated exotoxin, to use it as a vaccine to produce antibodies. In endotoxins, you would not find this ability of your immune system to make antibodies. The lethal dose for endotoxins is small, and for endotoxins a lot bigger. The representative diseases here are listed as gas gangrene for exotoxins, tetanus, botulism, diphtheria, scarlet fever. And for endotoxins, we have typhoid fever, UTIs, and meningococcal meningitis. Let's take a look at plasmids, lysogeny, and pathogenicity next. So plasmids are small circular pieces of DNA, and they can carry genes for toxins, or alternatively, of course, the production of antibiotics and other enzymes. and potentially a plasmid could produce a toxin that then causes toxic cellular effects. Lysogenic conversion, that would be change characteristics of microbe due to the incorporation of a prophage. So basically here we have pieces of genetic information that can cause the problems. And now next up, pathogenic properties of viruses. So viruses are always intracellular parasites, and so the visible effects of viral infection on a cell could be that we're stopping cell synthesis because cell lysosomes to release enzymes that could be toxic. A virus could also cause the creation of inclusion bodies in the cell cytoplasm, fusing cells to create a syncytium, that's possible. Typically, you're changing the host cell function to reprogram it to make more viruses. You could induce chromosomal changes in the host cell, and you can induce antigenic changes on the cell surface that then cause an immune response. You can cause cells to lose contact inhibition, and thereby leading to cancer. The cells sort of become autonomous. They don't want to be part of a tissue anymore. And viruses can also cause the production of interferons that then can be released from a virus-infected cell to protect and warn the neighboring uninfected cells. Here are some images that show you the cytopathic effects of viruses. Let's take a look first here on panel A. We have inclusion bodies that are being formed right here. And then here we have a cytoplasmic mass that has formed in this cell on panel B. And then over here we have a human fibroblast that was transformed by Rous sarcoma virus to become cancerous. It's all rounded up and doesn't want to be part of the tissue anymore. It has lost contact inhibition. It's on its way to be cancerous. Next up are fungi and some toxins that they can produce. Usually it will be toxic metabolic products that they can make. They can also provoke allergic responses, and they can produce toxins that inhibit protein synthesis, such as the trichothecenes. Proteases can modify host cell membranes, and capsules that they can have prevent phagocytosis. So here's some examples of fungal toxins. Ergot toxin, that one is pretty famous. It's an alkaloid toxin that causes hallucinations. Aflatoxin is a carcinogenic toxin produced by the fungus Aspergillus, highly carcinogenic, very carcinogenic. So aflatoxin is something to really look out for. Mycotoxins are produced by mushrooms, are neurotoxic. You have amanitin and phalloidin as an example right here. Some people actually enjoy taking these things because they can produce also hallucinations and get you on some trips. Next up, protozoa. It's usually their waste products that cause symptoms. They can avoid host defenses by digesting cells and tissue fluids, growing inside of phagocytic cells, and then they can employ antigenic variation, so it means they change their surface structure to trick your immune system. Helminths, so worms that is, they can cause damage to your body because they use the host tissue for their own growth. They produce sometimes very large masses and can cause thereby cellular damage. Some of them they produce waste products that are not good for you and that can even cause symptoms. Some algae can also be toxic. There are some that produce a neurotoxin called saxitoxin. An example for that would be paralytic shellfish poisoning. Next, I hear the portals of exit. Basically, those are the routes that a pathogen would use to leave your body and move on to the next victim, to the next host. So a lot of pathogens, they use your respiratory tract and they get out by coughing and sneezing, such as coronaviruses. Gastrointestinal tract is also a popular portal of exit. That will be used through fecal material and saliva also. The genitourinary tract, that will be urine and secretions from penis and vagina. The skin can act as a portal of exit, and then also blood. There will be arthropods that bite, or needle pricks, hypodermic needles. And here's our last slide summarizing the pathway of a pathogen through your body. So you have first, we're starting out with the portals of entry, and there's a list of them right here. Then don't forget about the ID 50, the infectious dose 50, so the number of invading microbes and how they attach to your cells, then how they penetrate or evade your host defenses, and there's the items listed here, how they cause damage to your host cells, the items listed, and then finally your portals of exit.